Today we're talking about Mendel's laws of inheritance. So here we have some pictures of families. We have Gwyneth Paltrow and her mom. Notice the eye shape and color, the cheeks. They look very, very similar. Here we have Brad Pitt, Angelina Jolie, and their child. And we also have the fork and the spoon. So when they cross, they might wind up with a slotted spoon, or they might wind up with a spork. Each child gets characteristics from the parents, but obviously they are not the same as the other child. By the end of this lesson, we need to be able to use genotypes to determine phenotypes and be able to explain the law of segregation and the law of independent assortment. So take a minute to look at this picture, see how it is showing genetics. Remember, genetics is the study of heredity, and heredity is the passing of traits from parents to offspring. Hopefully you're noticing that the bride's side of the family is much shorter than the groom's side of the family. I'd like to point out these ladies here are wearing flat shoes, while these two ladies here are both wearing heels. And they're still, even with heels on, much shorter than the groom's side of the family. So you can tell that one side of the family, the groom's family, is passing that tall trait along, and the bride's side of the family is passing that shorter trait along. A quick review, Gregor Mendel was a monk who lived in Europe. They sometimes call him the father of genetics because he conducted experiments with pea plants and found out that there are characteristics that are being passed from parents to offspring. He found out that the traits can be dominant or recessive, and he found that out by crossing these pea plants. He took a pure tall, which means it has two tall traits for the dominant trait, and a pure short plant, which would be two recessive traits. And what he found was that when he crossed them, they wound up being tall plants, which tells us that tall dominates over short because it's what shows up when you cross them. So these here would be capital T, lowercase t for each of the plants. They would have gotten a tall from the one plant, from the tall parent, and then they would have gotten a lowercase recessive from the short plant. When we cross these two hybrid plants, because they both have big T, little t, sometimes you get a plant that has two capital T's. Sometimes you get one that has capital T, lowercase t. This one also would be capital T, lowercase t, depending on which parent gave the uppercase and lowercase t. And then this one, which is short, happens because we get a lowercase t from each of the parent plants, and that recessive trait can finally show up in the phenotype. Now, Mendel's law of inheritance, there's two of them that we're going to talk about today. The law of segregation. This one talks about alleles, those different forms of the gene. So on the chromosomes, you've got two halves to it, each part of the chromosome. You've got the part you got from the mom and the part that you got from the dad. And along those chromosomes, there are alleles or genes, and you have the allele for each trait. So that gene, you're going to either have the dominant or the recessive trait. If we're talking about ones that just have dominant or recessive, remember blood type, there's actually three alleles, A, B, and O. But those alleles are paired up, one on mom's chromosome that was inherited, and one on dad's part of the chromosome that was inherited. And remember, those genes line up in the same spot, or alleles. So when they go through meiosis, they're going to pull apart randomly. So what we have here are 4 blanks that we need to fill in. These are words that we know from our reproduction unit. So it says allele pairs separate randomly or segregate from each other during the production of gametes. What are the 2 gametes that we talked about? Remember, there's one for the female and one for the male. And which type of cell division has that gamete production happening? We also want to fill in that the sperm or egg cell carries only how many alleles for each trait. Remember, we're getting half the number of chromosomes in the gametes through this process. Hopefully you said that there is the sperm and egg cell. Those are the gametes, and if you switched the position on those and had egg here, sperm here, that's fine. And then this all happens during meiosis, and we know that the sperm or egg cells only carry one allele for each trait because each parent has two alleles, one they got from their mom, one they got from their dad for a certain trait. And remember, those genes are on the same spot for each. This is widow's peak here. The widow's peak is going to be here from the other parent. And then each gamete gets one of these in the gametes because they're going to combine with the other parent to get the other set of chromosomes or that have the genes along them. So each gamete contributes its alleles, giving the offspring the complete set of chromosomes that it needs. So law of segregation. To be segregated means they're moving apart. And this is during that meiosis process. Just because they're lined up on the same spot on each chromosome, when those X's break apart, they're going to be randomly pulled apart. And what you get in each gamete is going to be a random process. The law of independent assortment basically just means that each trait that is being passed to the offspring through those gametes is going to be completely separate or independent of the other ones. So just because you inherited blonde hair does not mean you're going to have brown eyes. If there was a pea, that color that was green, that doesn't mean that you're going to have the round pea shape to go with it. If you're inheriting a widow's peak, that is going to be completely separate from having attached or unattached earlobes. So every trait that is being passed from parents to offspring is independent of the other ones. Just because you got one trait for something, one allele for something, doesn't mean you're going to get a specific other one to go with it. So what this means is our Punnett squares get very large very fast. So we have our Punnett squares where we're looking at one trait at a time. You've got mom's two traits, you've got dad's two traits. You fill it in to find out what possible combinations the babies have. If you're looking at two traits from each parent, now the Punnett squares are going to look like this. So we've got the pea color, and this is yellow, and yellow here. And then we have the green pod. So we have yellow seeds, green pods, and you have to fill in if this is the parent's genotype, and this is the parent's genotype. You have to take every one of the letters here. It's kind of like the FOIL method in math. You have to take this one and put it with this one and match it with that one. Then you have to take this other possible letter and match it with those other two possibilities for the other trait. So you have that on one side. So you've got big G, big Y, big G, little g, big Y, and little g, little y. You got to do the same thing with the other parent. And now we have 16 boxes that we have to look at. And it changes the probabilities because now instead of 4 boxes and each one being 25%, you have to divide 100 by 16 and then figure out the probabilities for each of these different outcomes. If you were to do another trait on here, it would get much bigger very quickly because each trait is inherited independent of the other ones. So just because you get a green doesn't mean for the pea pod, you're not going to necessarily have a yellow seed inside. Each one is independent of the others.